Good afternoon, everyone. My name is Arvind. I'm a faculty member in the Department of Bioinformatics, Radiation Oncology, as well as with the Cancer Data Science shared resource with the Global Cancer Center. And it's my privilege to speak to you all today about bioinformatics for the SCSAP community. Um, an example of where these kinds of technologies can be brought together is, for example, illustrated in the following graphic. Imagine if you want to ask a science question that brings together aspects of multiplex IHC, like what Tim described, single cell analysis, which is non-spatial, or a spatial platform like spatial transcriptomics or CyTOF or imaging CyTOF, and put that together with what you might have already discovered by way of hypotheses from bulk RNA-seq data. And so the question that we want to ask is, how do we build this kind of integrative bioinformatics infrastructure to be able to leverage spatial bioinformatics towards enabling science at the interface of all these multi-omics technologies? That is the goal of the bioinformatics pipeline we're trying to build within the SCSAP program. You've already heard a lot today about key enabling platforms like the Vectra Multiplex IHC platform, spatial transcriptomics platform from 10x and nanostring, etc. Others are coming through as well. Proteomics platforms like imaging CyTOF. All these are making lots of data available to us. And we now need analysis pipelines to be able to bring this data into useful information that can help hypothesis generation, validations, etc. So an example of the kinds of analysis that we can do is illustrated in this figure. This is work that's primarily being done with the Visium 10x platform, but you can see that once you get the data in the right kind of structure, doing these kinds of analysis becomes straightforward regardless of the kind of platform, and that's our goal. So with Visium 10x, however, this is what we can do. If you've got transcriptomic data at the spot level, not necessarily single cell, but spot level, you've got spatial data, you can look at spatial coherence in cell type location or gene expression, etc. You can also use single cell data by itself to be able to infer things like temporal coherence and cell state. Putting these together, having the spatial information available to you, you can ask questions like, what cell pairs tend to be nearby, right? Is there mutual attraction or mutual repulsion between different kinds of cells in the microenvironment? The spatial component coupled with the transcriptomic component also lets you ask questions about what genes are influenced by the existence of certain kinds of neighbors in the cells in the neighborhood. Finally, one can take all this together and ask conventional questions like pathway scores and GSCA, etc., but in a much more spatially sensitive manner. So we already built pipelines for these kinds of analyses with the Visium 10x platform, and this is work jointly with my students, Morgan, Vishwesh, Anand, who've been doing this for quite some time now, in collaboration with some fantastic labs here at UM. Now we've got a whole variety of analysis available. Like I said, you've got spot deconvolution for inferring different mixtures of cell types, like what you might see here. You can look at spatially aware clustering that brings both the spatial component and the transcriptomic component in together. You can answer questions about pathway enrichment and GSCA with pathway analysis, but in the spatial transcriptomic context. And once you've got these cell locations, you can now go and even do things like proximity maps, which I'll describe in a few minutes. And the point to mention is, as I said, the goal is to be forward-looking and be integrated with any and all platforms that are emerging in the infrastructure. They could be platforms like geomics, they could be up-and-coming platforms, they could be platforms like Merfish or cartography. And the idea is to be able to bring a set of harmonized analysis pipelines to this data once we figure out how to go from the acquisition instrument to a commonly usable data structure. That's been our primary work so far. But the goal is to be extensible to any kind of spatial transcriptomic modality looking for. Now I shift gears to Vectra Multiplex IHC. You heard a lot about it from Tim. So the kind of things that you can do are in two stages. One stage is where the software accompanying the instrument itself lets you do a whole bunch of analysis. For example, you can look at these images, you can unmix the colors, you can segment the cells, you can export the cells as files that contain the cell's location and the cell type. You can, once you've got that, you can look at differences in the population and abundance of these different cell types in the microenvironment. You can even go one step further and try to do some kind of um, simplistic rendering of the data involving things like t-SNE or UMAP to be able to get a better sense of what's going on. But because you've got spatial location, you can go one step beyond this phase. You can actually look at things like infiltration spatially as well as interaction. And what I mean by that is the following. Imagine in this case, you can see three different scenarios of variation in spatial coupling between the red immune cells and the cyan colored tumor cells. In this context, the infiltration between the immune cells and tumor cells is minimal. Here it's somewhat moderate. Here it's quite a bit. And so we basically come up with statistical techniques that lets one measure these things quantitatively. Once you've got these quantitative measurements, you can actually go ahead and do integration of those numbers with other numbers like clinical information or molecular data and build a multivariate model if that's what you want. So this aspect is about infiltration. The other one is of spatial co-location or spatial interaction. So you want to ask the question, is there an overrepresentation of cell type 1 within cell type 2's radius in the microenvironment? And so you can ask questions about attraction and repulsion between different cell types, their enrichment and depletion, so on. And as I said, once you've got these numbers, you can then link them with clinical data or molecular data to be able to ask questions at the scale of lots of slides. So here's an example where we did this for stratifying survival based purely on multiplex IHC data and those spatial infiltration curves for survival and prognostic modeling in pancreatic disease. You can use this not just for survival modeling, but also to, for example, aid with histological classification, like classifying low and high grade disease. We've done this kind of work in GI data. We've done this for in liver. We've done this for breast. We've done this for lung. And we've built models that let us even look at the extent of which different kinds of interactions might influence the model's predictive performance. So the key takeaways I want to leave you all with is that analysis workflows for vector multiplex IHC and 10x presumed spatial transcriptomics are already available and developed, and we are actively deploying them in different projects. So if you want to use those platforms, they are ready to use, and we'd be happy to help you. For nanostring geomics data and imaging site-off, things are a little bit more in development. But as I said, once you figure out a way to export the data from the native platform that accompanies these instruments, the analysis should not be that bad. So we are hoping that we can do the export of the data sets outside of the geomics DSP software or the Hyperion software to be able to get the data into a form that is amenable for analysis. And as I said, once you get these numbers, you can then do high throughput analysis by doing this on hundreds or even thousands of slides if you've got clinical data or molecular data that you want to be able to analyze. So it's just a matter of putting it all together, and we've experienced doing that. So if you think any of these platforms and ideas appeal to the kind of questions that you want to ask, please email us and we'd be happy to have a conversation around this and figure out ways to help you. In closing, I just want to quickly acknowledge the organizers for their kind invitation to speak today, Vanessa and Sunita. Also, the generous funding from the BSI has been instrumental in getting us up and running. Very grateful for the BSI in terms of building this platform. And this has definitely been a team effort. We've had help from leadership for visioning and lots of folks around methodology development, as well as collaborators to help us ask important questions. I want to once again acknowledge the help of Vishen Wu, Chris Gates from the bioinformatics core, as well as my own labs folks, Morgan, Vishwesh, Anand, and Santoshi, who worked very hard in building ready-to-use pipelines for bioinformatics analysis of these kinds of spatial data sets. In closing, thank you again for your time and attention, and please do email me if you have any questions about spatial analytics and bioinformatics. many of these platforms. We'll be very eager to hear from you and collaborate. Thank you again for your time today.